Hey everyone. So we left off class just talking about how sexual reproduction leads to genetic diversity. Okay. I really encourage you to make sure you go through both mitosis and meiosis. Make sure that you're understanding the differences between the two. This chart is one start, but also just making sure you understand the visuals, as you can see in these diagrams, how things are different. Again, using the terminology and the language in terms of what's happening at each of the stages, how the chromosomes are lining up differently. It's super important that you know this and understand it really well. So in terms of where we left off, that during meiosis, because of the way in which chromosomes are lining up as these homologous pairs rather than single file, we get this genetic diversity. So whether it is through independent assortment, where the chromosomes can line up differently, uh, leading to a variety of different gametes, or whether it's through crossing over of the sister, and sorry, I should say non-sister chromatids, so that's very important. So you're kind of think of it like one of your mom's chromosomes, the sister chromatid is crossing over with one of your dad's chromatids, right? So the non-sister chromatids. When that happens at these locations referred to as chiasmas, we have bits of one chromosome on the other homologous pair. So in this case, so you got bits of red on the blue chromosome here, and over here we got bits of blue. And you notice how they exist in the same position because of those chiasmas and the specific sites where they're crossing over. So that creates genetic diversity and why you don't look like any of your siblings unless you're identical twins. Now, there's some other things that can happen in terms of the action during metaphase and the pulling of chromosomes to opposite sides. Sometimes this can happen incorrectly, actually leading to damaging results for the daughter cells, in this case, our gametes, right? So when chromosomes don't separate properly, if chromosomes do not separate properly during metaphase, this can lead to an alteration in chromosome number. So today we were talking about karyotypes and how we can look at an image of an individual's chromosomes. These are karyotypes. I want you to pause the video and see if you can find the difference between the karyotype on the left here versus the karyotype on the right. So if you pause the video and looked carefully, hopefully you would have noticed that this karyotype over here is a normal karyotype having a pair of chromosomes, right, for each set, all the way down to both 21 and 22. There's 2 chromosomes for every number. But if we look over here on the right at chromosomes 21, there happens to be an extra chromosomes rather than there just being the pair, 2, there's a third. All right. And so this is what is called trisomy of autosomal chromosome 21. Now, something about our chromosomes, we talked the other day about our sex chromosomes. So here you can see that this person is a female because they have both X chromosomes. And similarly here, this person is a female as they have 2 homologous X chromosomes. But when we talk about our chromosomes 1 to 22, chromosomes 1 to 22, we refer to these as our autosomal. Remember somal, somatic, right? Autosomal chromosomes versus pair number 23, we specifically refer to as our sex chromosomes, right? And that's because although they contain other genes, like our autosomal chromosomes carry many genes for a variety of traits throughout our body. Of course, chromosome 23 being our sex chromosomes, they also contain genes that dictate other traits besides, our gender and our physical characteristics that make us male and female, right? Whether it's our organs, our reproductive organs, penises, vaginas, testes, ovaries, etc., as well as those secondary sex characteristics, broader shoulders, deeper face, deeper voice, more hair, etc. So they have certain characteristics that are non-gender determination, but also they are carrying the genes that determine our sex, primary and secondary characteristics. So there's some more definitions, basically, making sure we understand the difference between an autosomal chromosome and a sex chromosome. So in this case, because it's pair number 21 here, and that is an autosomal chromosome. So this is what is called trisomy. So 3 bodies, so 1 extra chromosome of the 21st pair. This is a process of non-disjunction. So non-disjunction process occurring during metaphase in which either homologous chromosomes I'll abbreviate that, or sister chromatids do not separate. And so we've got some pictures here. So we can see here in this case, we have normal chromosome separation in which this is dividing, and we have 2 chromosomes going this way and 2 chromosomes going that way. But over here, if we look, you've got both chromosomes going to the same side. Again, if we look over here, here's another one, non-disjunction in meiosis 1. This is in the case where you have normal meiosis 1 has taken place, but then you get non-disjunction in meiosis 2. So you can have it happen in either place. So here you're starting with a normal cell, but when the chromosomes separate, you can see you have both chromosomes going to, or one chromosome going to this side, whereas here you have a normal cell, right, where the chromatids are separating accordingly, but here the chromatids fail to separate. So this is occurring in meiosis 2. Here, the homologous pairs, this is occurring in meiosis 1, the homologous pairs fail to separate. And so both pairs are going to the same side. So here you can see this leads to an abnormal number of chromosomes in this grouping and an abnormal number in this grouping. When it occurs in meiosis 2, you can get a normal grouping where this one in this direction occurred normally, giving you here, again, we're starting with 2 pairs, so 4 chromosomes, but we separate to get 2, so that's normal. But on this side, these sister chromatids failed to separate, so we have 1 with 3 and 1 with 1. Neither of these have the normal number of chromosomes. Okay? So if I would review that, look at some pictures, diagrams, again, go through the definition, but ultimately non-disjunction occurs when either homologous chromosomes or sister chromatids do not separate properly, right? And when we're talking about homologous chromosomes not separating, that's like the first situation that occurs in meiosis 1. When we're talking about sister chromatids not separating properly, that's occurring in meiosis 2. Okay, now non-disjunction can occur in any pair of sorry, I had a sneeze coming on. Non-disjunction can occur in any pair of homologous chromosomes, autosomal or sex chromosomes. However, sex chromosomal abnormalities seem to have less impact on the overall genetic imbalance for a couple of reasons, right? So one reason this imbalance tends to have less of an impact if it occurs in our sex chromosomes is that the Y chromosome has relatively few genes. The Y chromosome is small with relatively few genes. And secondly, remember that females carry both X chromosomes, but in mammalian tissue, like any mammal, in mammals, usually they function with only one in mammals, and mammalian cells operate with only one X. All right, so what that means is that the cell might have both X chromosomes, but it mainly accesses one X chromosome for its protein production, right? And it's always accessing that same X chromosome. We'll see this and talk about this a little bit more when we get into chapter 9 in genetics and some interesting traits and characteristics that arise by operating on one functioning X chromosome. So we can see here's some non-disjunctions, variations in when our sex chromosomes don't segregate properly. And so, here's a situation in which there's no difference. This would actually result in a normal male, even though they've got two Y chromosomes. Here, again, same thing. You've got multiple X chromosomes. So this would result in a normal female, but it's more when there's this imbalance. Like here, you've got two X and a Y. This occurs in males, referred to as Klinefelter syndrome, and then XO, and the O meaning that they only have the one X chromosome. XO, what's going to happen here is some dividing cells may not end up with a complete chromosome, and so Turner syndrome occurring in females. Now, last thing, so this is what we call chromosomal mutations, right? Non-disjunction resulting in an abnormal number of chromosomes, and it can lead to a variety of different disorders or syndromes. Another type or a variety of types of mutations occur within individual chromosomes, and they're pretty self-explanatory here just by the names of them. So in this case, you can see that here's our normal chromosome, and what's happened over here is we're missing this section here titled B. So we call this a deletion, right? These are basically variations in alterations of chromosomes. They can lead to birth defects, cancer, and several types of diseases and disorders. Here in this case, you've got an extra copy of the B. So we call this a duplication, right? So if we want to put those in, so piece of chromosome missing, right? We have piece of chromosome added, so additional here. And then other things that chromosomes can do is what are called inversions, in which the regions, almost like crossing over, but it's occurring on the same chromosome. And what it's done is it's reversed the genetic inversion. And this can be important because ultimately genes that border one another can be connected in a certain way, right? So regions of chromosomes switch places. And so that can create problems in terms of what neighboring genes are brought or not brought in that inversion. And here we have a translocation, and this is between two non-homologous chromosomes. So this is not like crossing over where it's two homologous chromosomes. This is like crossing over between non-homologous chromosomes. So like let's say this is chromosome number 3, let's say, and this is chromosome number 7, right? So crossing over between non-homologous chromosomes. We'll talk more about this when we get into our topic on genetics, but all of these are alterations to chromosome number and chromosome shape and function, crossing over between non-homologous chromosomes. All right, I will likely be posting more videos in Chapter 9, so stay tuned for some emails regarding notes you should be doing. I'd like to free up some class time next week to work on some other projects. All right, see you tomorrow, or not.